Hello everyone and welcome to the another session of the lecture series, and today's topic is cell cycle and cell division. And this lecture, what we will doing, we'll be discussing and solving the NEET previous year questions. Alright, and this lecture is in the continuation of the previous lecture, which was again based on the NEET previous year questions. So let's start with the first question. So here you have been given this figure, okay? So this is another type of MCQs, alright? So here they have given a stage in the cell division, alright? And you have to, what you have to do? You have to select the answer from the following options and you have to identify the stage, alright? So okay, so options are anaphase, telophase, equatorial plate and the kinetochore. So what should be the answer? You can see here we have the aster. Okay, what are aster? They are nothing but star-shaped structure, okay, and consist of microtubules that arise from the centrosome. Alright, this you can say is the nuclear membrane. Alright, this is the chromosome material, okay, and the two cells are, you can see, are dividing, alright, and the chromosome have reached to the opposite poles, or the genetic material, okay, have reached to the opposite poles. So what should be the answer? Answer should be telophase, okay, so this was telophase. Now coming to the next question. Now here they are asking during meiosis 1, okay, during meiosis 1 the chromosomes start pairing at which stage? So options are zygotine, pectine, diplotene and leptotene. Alright, these are the stages of prophase 1, okay. So here they are talking about the prophase 1 of meiosis 1. Alright, prophase 1 of meiosis 1. Prophase 1 of meiosis 1 is divided into leptotene. Okay, in leptotene what happens? Condensation of chromosome takes place. Okay, condensation of chromosomes takes place during leptotene. And the chromosome are clearly visible. Then what happens in zygotene? Synapses, or you can say pairing of chromosome takes place. And what they are asking? Chromosomes start pairing. This is the question. Alright. Now in pectine what happens? Crossing over takes place during pectine. Okay. Crossing over. During diplotene what is seen? Chiasma or chiasmata formation is seen during diplotene. Alright, leptotene we have already seen what happens during leptotene. So what should be the answer? Answer should be zygotene. The pairing of the chromosome, which is known as synapses, is seen during the zygotene phase of prophase 1. Now coming to the next question, which is, here they are asking during the metaphase stage of mitosis. Okay, spindle fibers attached to the chromosome at, okay, so what happens if this is a chromosome, okay, centromere is present here, and what does centromere have? Okay, centromere have kinetochores in them, okay, so here kinetochores are present in this region. This is a centromere, and within centromere there is an area, okay, where spindle fibers attach, and that is known as the kinetochore. Alright, so first option is kinetochore, second is both kinetochore and centromere. So here they are confusing you with kinetochore and centromere. Third option is kinetochore and centromere and the area adjoining the centromere. Alright, and the fourth option is just the centromere. then what should be the answer? Okay, according to this, answer should be kinetochore. The spindle fiber attached to the kinetochore present in the centromere of the chromosome. Now coming to the fourth question, here again they have given you this figure, and what you have to do, a stage of mitosis is shown in this diagram, and they are asking which stage is this and what are the characteristic of this stage. Okay, and they are talking about mitosis here. Alright, so here you can see again, chromosomes are aligned here at the equator. Okay, or the equatorial plate. They are aligned at the equator. Okay, or you can say at the metaphase plate. They are aligned at the metaphase plate. Alright, so here are the options: anaphase, okay, then late prophase, metaphase, and metaphase again. Alright, so what should be the answer? Answer should be, this is the metaphase stage, and what are the characteristics? Chromosome move to the spindle equator, means the metaphase plate or the equatorial plate, and chromosomes are made up of two sister chromatids. Okay, remember that chromosomes are made up of two sister chromatids. Again, this is a chromosome. Alright, so these two are known as sister chromatids. A chromosome consists of two sister chromatids. Okay, so this is the answer, option D. Now next question is spindle fiber attach on. Again, this is a similar questions, okay, but I have included this because the options are different. Alright, so these are our options. Centromere again of the chromosome. Kinetosome they are asking, okay, not kinetochore, okay, of the chromosome. Third option is telomere of the chromosome. Fourth option is kinetochore of the chromosome. Okay, so the answer should be option D. Spindle fiber attached to the kinetochore of the chromosomes. Now coming to the next question, here they are asking in meiosis. Okay, so during your exam what you should do, you should highlight all these words first. Okay, in meiosis, second keyword is crossing over is initiated at zygotine, diplotin, pectine or leptotine. So again they are talking here about the prophase 1. Alright, of meiosis 1. So zygote again what happens? We already know synapses or pairing of chromosome takes place. So what happens? During diplotin what happens? Chiasmata formation is seen. During pectine, crossing over takes place and recombination is taking place. During leptotine, chromosome condensation is taking place. Alright. So what should be the answer? Answer is crossing over takes place during the pectine stage. Now coming to the question 7. Here we have what is the question? Which of the following is not a characteristic feature during mitosis in somatic cells? Mitosis we know takes place in the somatic cells or vegetative cells which are diploid. Alright. So here they are asking which is not a characteristic feature. Alright. So first is chromosome movement. Second is synapses. Spindle fibers is the third option. Fourth option is disappearance of the nucleolus. Alright. So chromosome movement takes place during mitosis. Spindle fibers also are formed. Disappearance of nucleolus also takes place. Okay, at the end of prophase. So option B is the answer because synapses occurs during meiosis and not mitosis. Okay. So answer should be synapses. Now coming to the next question. So question is which of the following options gives the correct sequence of events during mitosis. They are asking about mitosis here. So here are the options, all right? You can take a look at the options. Condensation, then nuclear membrane disassembly, arrangement of the chromosome at the equator, centromere division, segregation, and then the last stage, telophase. All right? I'll give you some time to look at the options. Okay? Now what happens during mitosis? This is the answer. All right? First, condensation of chromosome takes place. Okay? Which is happening during the prophase. Okay, condensation of chromosome takes place during the prophase. Then nuclear membrane disassembly. Okay, again at the end of prophase. At the end of prophase, we see the nuclear membrane disassembly. Then arrangement of the chromosome at equator. This is seen during the metaphase. Okay. Centromere division. This takes place during the anaphase. Segregation of the chromosome again takes place during anaphase. And the last stage is telophase. The mitosis is divided into prophase, then we have metaphase, then we have anaphase, and then we have the telophase. And after telophase, we have cytokinesis. So this is the correct order of mitosis. Now next question is, during the cell growth, DNA synthesis takes place. Okay, in which stage? So here they are asking about, or they are talking about interphase. Interphase is a stage before prophase. Okay, so if we talk about cell cycle is divided into interphase. Okay, and then we have the M phase or the mitotic phase. Okay, interphase is further divided into G1 phase. Then we have the S phase and we have the G2 phase, and then is the actual cell dividing stage known as M phase. Alright, so options are S phase, G2 phase, G1 phase and M phase. So DNA synthesis takes place during the S phase. Okay, you can say DNA synthesis or DNA replication. S phase is also known as the synthetic phase. Here the DNA replication takes place as well as the division of the centriole takes place. Alright, so coming to the next question. When cell has stalled DNA replication fork. Okay, what does this mean? Stalled DNA replication fork means there is an hindrance or there is a blockage. Okay, when there is blockage of the replication fork means there is no replication. Alright, then which checkpoint should be predominantly activated? Okay, so options are G1S, G2 or the M checkpoint. We have the mitotic checkpoint, or they are asking both G2M and mitotic checkpoint. Okay, so let me first give you some background about this. So there are 3 checkpoints during the cell division. First is the G1 checkpoint, which is at the transition of G1 and S phase. During this, what happens? If the DNA is damaged or if there is damage in the DNA bases. Okay, if the DNA bases are damaged, then what happens? This checkpoint will repair the damaged DNA and then only it will allow the cell cycle to get to the next stage, that is the S stage. Okay, so it repairs the DNA which is damaged, okay, and then allows it to get, okay, to go to the next stage. Second checkpoint is the G2 checkpoint. In G2 checkpoint, what happens? It is at the transition of G2 to M phase. All right. So here what happens? If the DNA is incompletely replicated, okay, so this checkpoint checks if the DNA is incompletely replicated. Or if the DNA is damaged, okay, then this checkpoint is activated and it won't allow the cell to go to the next stage. All right. Third checkpoint is the M checkpoint or the mitotic checkpoint. So this checkpoint is present at the metaphase and the between metaphase and anaphase. Okay. All right. And what does this checkpoint do? This checkpoint checks if the spindle microtubules, okay, if the spindle microtubules are attached to the sister chromatids or not during the metaphase. If they are not attached, if the spindles are not attached to the sister chromatid, then there will be no separation in the anaphase. Okay, so it helps in the transition also from metaphase to the anaphase. So what should be the answer? They are asking about here the incompletely replicated DNA. So answer should be option B. So this G2M checkpoint should be activated if there is a stalled DNA replication fork leading to incompletely replicated DNA. All right. Now the next question is, here we have to match the stages of meiosis. This is another type of question, okay, in column 1, okay, and with the characteristic features in the column 2. All right, so here are this is the column 1, column 2. Pectine first we have. Pectine what happens in the pectine stage? Crossing over takes place. All right, so A should be third. Metaphase, what happens? Chromosome aligning at the equatorial plate. Then B should be fourth. Diakinesis, terminalization of chiasmata takes place. Alright, so C should be second. And zygote, in what happens? Pairing of homologous chromosome takes place, which is known as synapsis. So D should be 1. Alright. So these were your options. Okay. So correct answer is this one. Alright. Now moving to the next question. Here we have to, what they are asking, what is the correct sequence of phases of the cell cycle? Cell cycle is again divided into interphase and the M phase. Interphase consists of G1 phase, S phase and G2 phase, and then we have the M phase. Okay, so the correct sequence should be which one? First we have the G1 phase, then we have the S phase, we have the G2 phase, and then in the end we have the mitotic phase, alright, where the actual cell division takes place. From G1 to G2, it is interphase, and then we have the mitotic phase. The next question is cells in G0 phase they are asking. Okay, G0 phase is known as the quiescent stage phase or the inactive phase, where the cells are metabolically active but they are not dividing. Alright, so options are: cells in G0 phase suspend from the cell cycle, terminate the cell cycle, exit the cell cycle, or enter the cell cycle. Alright, so what should be the answer? Answer is cells in the G0 phase, they exit the cell cycle at G1 phase and they enter the G0 phase. Okay, we have the G1 phase, we have S phase, we have G2 phase. Cells such as heart cells, okay, some skeletal muscle cells, lymphocytes, some nerve cells, they exit the cell cycle at G1, okay, and they enter the G0 phase. Okay, and then so this should be the answer: exit the cell cycle. The next question is the stage during which separation of paired homologous chromosome begins. Okay, zygote, there is pairing of homologous chromosome. Okay, here they are asking about the separation of the homologous chromosome. So this should be not the answer. Okay, Pachytene, what happens? Crossing over is taking place during Pachytene. Okay, so chromosomes should stay together, so there should be no separation. So this is also not the answer. Diplotin, what happens? There is chiasmata formation, okay, and there is separation of the homologous chromosome. Okay, there is separation of the homologous chromosome. And what happens here? You can see the formation of chiasma or chiasmata. During dikinesis, what happens? There is terminalization of chiasmata. So what should be the answer? Answer should be diplotin. Okay, separation of the paired homologous chromosome begins at diplotin. Next is a bit tricky question. Here what they are saying anaphase promoting complex means it is a complex, okay, which helps in the transition of the chromosome from the metaphase stage, okay, to anaphase. It helps in the transition of the metaphase chromosome to the anaphase. Alright, so here are the options. Okay, so let's look at the question again. Okay, anaphase promoting complex is a protein degrading machinery necessary for proper mitosis of animal cell, they are saying. Okay, if APC is defective in human cell, which of the following is expected to occur? Okay, so options are: chromosome will fragment, chromosome will not segregate, recombination of chromosome arms will appear. Fourth option is chromosome will not condense. Okay, I've already told you APC is a protein degrading machinery. It is a protein degrading complex, or you can say machinery, okay, that will attach to the chromosome present at the, or you can say they attach to the metaphase chromosome, okay, chromosome which are lying at the equator. And what they do? They attach at the metaphase chromosome and they help in the segregation of chromatids. Okay, so what should be the answer? Chromosome will not segregate. Okay, alright, so this should be the correct answer. Alright, This is the correct answer. Chromosome will not segregate if APC is defective in human cells. Now, here what you have to do, you have to match the following with respect to meiosis. Alright, again they are talking about the prophase here. These are the stages of prophase. Alright, in zygotine, okay, what happens? There is synapsis or the pairing of chromosome. This is a similar question, we have already done that. Pectine, what happens? Crossing over takes place during pectine. During diplotin, what happens? Chiasmata formation is seen, and dikinesis, there is terminalization of the chiasma. Okay. These are the options. You can have a look at the options. You can take your time. What should be the answer? Answer is this one. Alright. A 4, B 3rd, C 2 and D 1. Now next question is dissolution. Okay, or you can say breakdown of the synaptonymal complex occurs during. Alright, again they are talking about prophase here. Prophase 1. Diplotin, zygotine, pectine and leptotine. So this is very straightforward question. Answer is diplotin. Dissolution of the synaptonymal complex occurs during diplotin, okay, which helps in the separation or the separation of the chromosomes, and chiasmata is formed. Alright, now the next question is, here this is another type of question. Here we have to identify the correct statement with regard to G1 phase, okay, or the gap 1 of the interphase. So first is cell is metabolically active, okay, but does not replicate its DNA. Second option is DNA synthesis or replication takes place during a G1 phase. Third option is nuclear division takes place. Fourth option is reorganization of all cell components takes place. Okay, so during G1 what happens? Cell growth takes place. Okay. Second is cell is metabolically active. Cell is metabolically active. There is synthesis of macromolecules. Okay, lot of proteins are synthesized, lipids, okay, RNA is synthesized. Okay, so what should be the answer? Cell is metabolically active but does not replicate its DNA. DNA replication takes place during the S phase. Alright, so during G1 there is no nuclear division. DNA synthesis again takes place in the S phase. There is no reorganization of all the cell components. So answer, option A is the answer. The next question is fruit fly, okay, they are saying has 8 chromosomes, okay, in diploid condition it has 8 chromosomes in each cell. During interphase of mitosis, they are saying if the number of chromosome at G1 is 8, what would be the number of chromosome after the S phase? Okay, we have the G1 phase, S phase, G2 phase and the M phase. So at G1 chromosome number was 8. Okay. And what happens when the cell enters the S phase? There is the DNA synthesis or the DNA replication. Okay, if here the DNA content was suppose 8C, after the S phase, what will happen? DNA content will be double. It will be 16C. Alright, but the chromosome number remains the same, means it will contain 8 chromosomes only. Alright, again, let's take one more example. If the organism is diploid, means it contains 2C DNA content. Alright, this is at the G1 phase. After the S phase, what happens? There is DNA replication and still the chromosome number will remain the same, but the DNA content will be 4C. So chromosome number remains same. So the answer should be option B. The chromosome number will remain the same. It will be 8. Now next question is, which stage of meiotic prophase shows terminalization of chiasmata as its distinctive feature? This is also a straightforward answer. The answer should be, which should be the answer? Diakinesis. Pectine crossing over is seen during pectine. Okay. Zygotine synapsis is seen, means pairing of chromosomes. Leptotine, condensation of chromosome. And the chromosomes are visible. Concentration of chromatin, you can say. Diakinesis, terminalization of chiasmata. So answer is should be option D. Now next question is, the centriole undergo duplication during which stage of the interphase they are talking about now? Okay. Again, interphase we have G1 phase, S phase and G2 phase. And then we have the mitotic phase. Alright. So S phase, what happens? DNA synthesis takes place. And centriole division also takes place. In some animal cells, centrioles are self-duplicated. Okay. G1 phase I've already told you. Cell growth is taking place. Synthesis of macromolecules is taking place. Alright. During G2 phase, what happens? Final preparation for the cell division is taking place. Final preparation for cell division is taking place. There is duplication of the cytoplasmic organelles. Duplication of the cytoplasmic, or you can say cell organelles, such as endoplasmic reticulum, Golgi complex and all. So what should be the answer? Answer should be S phase. Okay. Centriole undergo duplication during the S phase. Now the next question is, again, it's a bit tricky. DNA replication in bacteria, okay, occurs, options are within the nucleolus, prior to the fission. Alright. Bacteria is a prokaryote and they divide with the help of fission. Next option is just before transcription. Last option is during the S phase. Alright. So in bacteria, what happens? DNA replication takes place with the help of mesosomes. Now what are mesosomes? Mesosomes are infolding or invagination, okay, of plasma membrane. It is an infolding or invagination of the plasma membrane, and they help in the, you can write replication, okay, replication of the nucleoid. Prokaryotic cells contains nucleoid, okay, and it also helps in the attachment of nucleoid with the plasma membrane. Help in the attachment of nucleoid with the plasma membrane. Alright, now what is a nucleoid? What is a nucleoid? Nucleoid is a irregularly shaped structure, alright, present within the prokaryotic cell, which consists of all or most of the genetic material. Most of the genetic material in a prokaryotic cell. Okay, so what should be the answer? Answer is option B. DNA replication in bacteria takes place prior to fission with the help of mesosomes. Alright, so answer is option B. Alright, so this was the end of this session. These are all the important questions, previous year questions. Alright, so focus on them and thank you very much for listening.